good around here.
Oh, good. Well, that's
I started
not
So I'm just going to start by making.
Today I'm going to introduce you to the soil food web. But before I do that, I'd like to share with you two things that you're not likely to hear from your major agricultural universities, let alone from your fertilizer salesman. The 2 points are this. Number one, the parent material of the soil in your garden or on your farm contains all of the minerals that your plants require to grow, to be healthy and reproductive. Point number two, bacteria hold the highest amount of nitrogen in their body, along with many other important nutrients that your plants require in order to grow. The caveat to these 2 points is this. Without a thriving, beneficial soil food web, your mineral component and your nutrient component will not be available to your plants. At this point, you will have to add whatever you can dream up that your plants might be lacking into the soil. So let's start talking about the soil food web. The soil food web is a diverse group of organisms that through their predator-prey relationship, they make nutrients available to your plants. That means by eating one another and pooping out and juices flowing out of their mouth after they've been eating, the nutrients are going into your plants and your plants can absorb nutrients as they need. In the image you can see that here we have plants and underneath it you have organic matter, which is basically anything that was once living but it's not living anymore. Your plants need what's in the organic matter, but they don't have the enzymes to break that down. So it requires bacteria and it requires fungi to break down the organic matter and turn it into something new that your plant can absorb. But the fungi and the bacteria don't have a way of making nutrients available in the way that your plant needs to absorb them. So then we require the protozoa, the nematodes, the microarthropods, and the macroarthropods to do this nutrient cycling. Let's take a look at the next image. Here you can see a beneficial fungal hyphae strand. This is a fruiting strand. It has wide diameter. It's the color brown. You have a mineral up in the upper right-hand corner, and you have lots of diversity of bacteria. This is a sign of a thriving soil sample. The diversity of bacteria is essential here because certain bacteria thrive in certain times of year more than others. But nothing's going to be available to your plants unless something can come along and consume them. And so this is where we have the protozoa and the nematodes and microarthropods coming in. This is an image of a testate amoebae. He has a huge mouth, he's got lots of bacteria in his belly, and also if you take a look at the rest of the slide, we have a diversity of bacteria around for him to consume. In the lower left-hand corner, you have a large fungal hyphae strand that if I could scan out, you would be able to see the diameter and the length on the strand, meaning we have phosphorus, magnesium, calcium, sulfur, all of these things available to your plants in the form that your plant needs, as long as something can come along and chew up this fungal strand. Here we have an image of a nematode. This particular one is a predatory nematode. It eats other nematodes, again transferring nutrients into a higher succession plant available form. There's all different kinds of nematodes and we identify them based on their mouth parts. So not all nematodes are bad, even though they tend to get a bad rap for that. This next image, we have a microarthropod. These guys are shredders. They come through, they break down the organic matter in the soil, they make it easier for bacteria and fungi to digest and break down. By default, this particular one could come along and chew his way through a fungal hyphae strand, making nutrients available to the plants. Here in this film, we can see a stalked ciliate consuming bacteria, releasing nutrients in a plant-available form. If you had many of these, it would indicate an anaerobic environment. Fortunately, in this particular sample, we just had one. So what I'd like to share with you is this. Your plants release something called exudates, and it's released through their surfaces, and it's released through their roots. Organisms, bacteria and fungi, are attracted to these exudates because they contain sugars, amino acids, and minerals. So your plant is calling bacteria and fungi to it by putting out the food that that organism wants to eat. So when you have the predators present in the soil, they are called to the root of your soil, and they consume the bacteria and they consume the fungi. So now you have nutrients being released exactly where you want them to be. So the organisms are doing this, and we don't have to apply synthetic fertilizers, fungicides, pesticides. If we do applications of fungicides and pesticides, we lose the soil food web. So let's talk quickly about what the benefits of the soil food web are. The soil food web creates soil stabilization. Organisms in the soil create the structure in the soil that your plants need in order to get their roots down. They create highways and byways through the soil so that water can percolate down, air can come down in at the root of your plant. And so now we have great soil structure happening, we have capillary action happening, so water is likely to be able to hold in your soil. Organisms are 70% water, holding water in your soil. When the predator comes along, it consumes the bacteria, releasing a bit of water along with minerals and nutrients available to your plant. We also have nutrient retention. Like I mentioned, the organisms hold the nutrients in their bodies. We have nutrient cycling through the predator-prey relationship of the organisms. We have plant fertility because your plants are getting what they need when they need it. And we also have carbon sequestration. Organisms need carbon in order to live. So by consuming one another, they store carbon in their bodies. The more organisms we have in the soil, the more carbon we're holding. So we have all of these benefits, these 7 benefits of a balanced system, a healthy soil food web. So now it's time to learn about how we create an environment for a healthy soil food web. When building the thermal compost pile, we've had great success with prepping the materials 3 days before the compost pile gets built. So what we do is we take our wood chip or our woody brown material and any other material like hay that would be dried out, and we water it every day for 3 days before we build the pile. That's not to say that we mix them together, because then they would start the decomposition process. So we make sure to keep our piles separate. Here you see we have a wood chip pile. Today will be the 3rd day of us turning and watering it. And you can see that it's getting nice and hydrated in the middle. The importance of watering the ingredients comes from the fact that as organisms dry out, they create a coating around their bodies. It's to help protect them from the environment. And that shell that they protect themselves with is really difficult to get rehydrated. So we find that it takes about 3 days to get that shell softened and get those organisms awake and ready to go again. Now when we build our thermal compost pile tomorrow, the hay will be able to absorb the water because the organisms have broken down and they've released themselves from the cellulose material of the hay or the wood chips. Over here I'll show you the hay. So this is the 3rd day of watering. This hay has a tendency to clump together so that oxygen can't get through, and that's one of the reasons why we don't just water from above. We turn this and water it. This is starting to heat up, so it is starting a bit of its own decomposition process. Turning and adding oxygen will keep it from potentially igniting into flames, but it also keeps us from developing too much actinobacteria. So it's important to remember what the difference between hay and straw is. Hay has sugars in it, which is a bacterial food. Straw, the plant sugars have left the plant, and so all you're left with is cellulose. So we're going to flip this and just lightly water it, but really today we're focusing on adding oxygen to this material. Yeah, that's perfect.
light on the grass, eh?
Light on the grass?
Well, it's already hot. I mean, it's breaking down. It's already higher in nitrogen.
Yeah, some of it is. Some of the wheatgrass.
It's hotter.
All humanity.
Sandwiches, they're so good. When I was in class, it made me look forward to Mondays. So I was like, no, I know it's Monday. Close the loops on our waste. Every time you put it in the dumpster, you put in the shredded cardboard. I don't know, there's a way that we can figure it out. It's just
Oh, there is.
convincing people that it's a worthwhile thing to do. And as soon
So by adding more green material, that's, I mean, the nitrogen-rich compost.
I mean, I don't know about that per se. I mean, the organisms, sure, it takes a really long time to break down. It's kind of like there's nothing
Added in with the middle component of soil.
like this. So the reason why we want the temperature is because we want to kill weeds.
Right. I mean, it's an idealized kind of
That you're smelling. But I'm kind of confused about that.
smell of rain.
Yeah. Like, when we do it inside, the piles get too hot and the worms have to leave. Come on in.
And it'll, once it starts going, it'll totally shrink. Yeah. And it's an Apple gift card,
Oh,
so I can't like exactly.
and that's why the school just had to switch. The things that it'll do compared to Final Cut
Yeah.
are amazing.
Growth or whatever.
When I do temperature readings on my compost pile, I take 3 different temperatures so that I can find an average temperature. Right now I'm determining where the middle of my compost pile is. I point the end of the thermometer at the middle, and then where the edge of my cage is, I put my fingers there. Then I look to make sure that I'm putting the thermometer in at the middle of the pile, so I'm looking to the top and the bottom. And I reach the thermometer in until my fingers reach the cage, and then I know that the tip has reached the middle of the compost pile. So I'll put in my last thermometer here. You want 2 to be in at the side and 1 to be coming down into the middle from the top. Right now I'm finding the middle of the compost pile this way, so when I hop up onto the cage, I know how far to put the thermometer down in. Once you have your thermometers in, it's going to take a few minutes before you have an accurate temperature reading. This is an ideal time to do your moisture readings. Taking moisture readings from your pile, you're going to want to take 3 different moisture readings so that you can find an average moisture. And the ideal moisture would be 50% saturation, so you're going to reach down into your pile as far as you can. This pile is pretty hot, so I actually can't reach down very far. This is something worth noting in your notes, how far down you're able to reach. So I squeeze, notice the water coming through between my fingers. I'd call that 50% moisture. So I'll put that back in the hole and cover it up. So you're gonna wanna take these readings from 3 different areas of your pile. If the cage allows you to take readings from the middle and even towards the bottom, it's a great idea. Just put that back in there. Again, I'm getting water pooling in between my fingers. 50% moisture, it's perfect. We haven't had to water this pile but once in all of the turnings that we've done for it. For more information about building a compost pile and temperature and moisture monitoring, go to my blog called the Microherders Manifesto.
What was this thing temp at?
The average was 163. We gave it a few more hours. We'll be right at our turn now. And we'll just pull it right around over there. Between the top and as we start getting down into the middle. So we're just going to set the top aside. Maybe we could do it right here. And then we'll start working in taking away the middle and filling in the outside edges of the pile. And then we'll take the outside of that and fill it into the middle of the new pile. Once we get to about the middle of this one, then we'll add the top into the middle of the pile. So you can actually see this ring right here. This is an actual separation between what's cooking in the middle and the outside layer. Smell a little bit of ammonia, which means that we have a bit of anaerobic conditions going on. We're losing nitrogen in the form of a gas. So it's really good that we're turning right now because we want to get air into the pile. Our organisms. So this is good. This is more than enough of the top. So now we'll just start scooping away in the middle, and we start building a layer around the outside. Fills as you drop them in so that they can get some air. Sometimes, like working with big strong guys, I'll just take a big shovelful and plop it in, but it's good to kind of shake the material so we're really getting air in there. And Andy, as you'll notice, like this side kind of wants to come away now, so instead of losing it on the ground, I'm just going to grab it and pop it in the middle. It's so great to have help turning piles. I'm a lucky lady. So as our middle starts to get up a little bit taller, I just lightly pull it out to build a base. I try to still keep everything more in towards the middle, so I try to not pull it out too far to the edges. So after that one, we'll start taking from the middle and build up the outside so we can add the top to the new middle. This morning we're going to make humic acid. So I start with a spaghetti strainer and a rack to put over the top of the bowl that I'm going to be catching the humic acid into. So first I'll just grab a couple handfuls from my finished compost pile. The amount of compost that you're going to need to make homemade humic acid really depends on the humic acid content of your compost pile. You can see that the color of this compost is about 70% cocoa. It's a nice kind of chocolatey brown color. It's not black, it's not light tan. So this means that we don't have to use very much to get the humic acids out. So just a couple handfuls is good. And I mound it up on the sides, and I make a little bit of a hole in the middle because that's where I'm going to be lightly pouring my water through. I want the water to slowly filter through the compost. What will happen is that as the water goes through the compost, the humic acids come through and go into the water. Any chemicals like chlorine or chloramine will get attracted to the humic acid because it has so many receptor sites on it, and so it won't take away the chlorine or the chloramine, but it'll basically make it inactive into the water solution. So now I'll start pouring the water in. This is city treated water. I just got it from the faucet inside. And notice I'm not dumping it all in there, I'm kind of lightly pouring it through. Depending on your quality of compost, you can make about 4 cups to 6 cups of humic acid from this amount. But really what I'm looking for is a similar chocolatey brown color in the water. So now you can see that the humics have come through, and now that they're suspended into the water. So this humic acid solution that I've made is going to be good for adding to compost tea before you make your compost tea. It's good to add to your extract before you make extracts. And today we're going to be using it to water our compost piles and also to prep ingredients for our compost piles, because we use a hose that is for municipal water.
So I'm just going to start by measuring out a pound.
Nice. So when we're making compost extract, I'm starting with a 5-gallon bucket filled just below the top so that I have room to massage the compost into the bucket. I've treated the bucket of water with humic acid, and I put 2 pounds of compost into a paint strainer bag. I know that I'm using 2 pounds because I've made extract with my compost before. When you're making extract for the first time, you're going to want to start with 1 pound of your compost, put it into your paint strainer bag, submerge it into your humic acid treated water, and you're going to massage vigorously for 60 seconds. When I have this bag submerged into the water, I'm rubbing on both sides of the bag. I'm pulling the bag apart. I'm getting my hand in where the compost is lumped together, and I'm pulling it apart. And I'm just massaging it very vigorously in the water. Extract is the soluble nutrients and organisms that are on the compost, suspended in water. So we're not adding any foods, we're just getting the soluble nutrients and organisms off of the compost. So it's been about a minute. Now I'm going to take a sample and I'm going to look at it through the microscope. Depending on what you want to grow, you're going to want, for like your vegetable garden, you're going to look for 3 strands of fungi per 5 fields of view. You're going to make a note of bacterial diversity, and you're going to scan for nematodes. This is day 5 of the Tolid tea recipe, and I'm going to check out what's happening in here through the microscope. So I'm going to take 1/4 teaspoon of the tea inoculate and mix it with 4 parts water. And we're just going to do a quick visual assessment to make sure that I have good fungi and that I don't have any weird organisms. This tea won't be done until 11 tomorrow. So I'm going to go ahead and add some humic acid to this solution. And so this humic acid is homemade. If you're buying humic acid online, you'll really only need to use about a teaspoon to 1 tablespoon of humic acid. Now, I also added about an hour ago 1 tablespoon, which is about 1/2 ounce of homemade fish hydrolysate. And then I add that in there about 1 hour before I put in my compost tea inoculant into this solution. So when I add the soluble seaweed, I'm going to put in about 1 ounce. So that's equivalent to about 2 tablespoons. So this stuff can get really sticky. So I add it into a container. And then I put some water in it and I mix it up to get it dissolved. Because otherwise, if you just drop it right into the bucket, it's going to become this like globby mess at the bottom. And you really want this solution suspended in the water. The soluble seaweed is a platform for fungi to attach themselves onto and digest. Okay, so at this point, I'm going to pour this back into my tea brewer. Rinse out all the goods. So now I'm going to take my inoculant into my tea bag and I'm going to crumble it up. I like to do that while it's in the tea bag so I don't have to touch it too much. It's a little bit sticky. This way, you don't have a big solid mass in your tea bag, and when I drop it into the brewer, the bubbles from the brewer are going to be pushing the solution around and getting out all the little guys that have developed little aggregates around them and get them suspended into the water. So now I'm going to drop the tea bag into the brewer and attach it. I've got a little clip on it here and I'm going to attach it onto this top wire part here of the brewer. And I stick my hand down in to make sure that it's moving about and getting air all in through it. So now we're going to let this brew for about 24 hours. Notice that we have the brewer outside. If it was a hot sunny day, I would want to be coming back every 2 to 4 hours to check the organisms in the tea. On hot days, the organisms tend to reproduce more quickly. So we have a cloudy day, it's going to rain. I think it's going to be okay to leave it for 24 hours and we'll come back and we'll check it tomorrow. Adjusting it to pull it up out of the solution a little bit.
Okay.
When we're applying compost to a piece of land, an important ratio to keep in mind is 1 ton per acre. 1 ton of compost per acre. So what I'm doing here, I've broken down my ratio for a 4 square foot area. It means that I would use 1/2 cup per square foot of soil, per square foot of earth. So just put down my compost, mix it in. Now my plants are ready to go in. These squash seeds have been soaked in compost extract overnight, and now they're ready to go directly into the soil. Soaking seeds in extract ensures that you have organisms coating your seeds so that when your seed begins to sprout, all of the roots will be covered in the organisms. Dipping plant roots in extract is also a great way to ensure that the root zone has an association with the microbial life. The ideal time of year to do a compost extract soil drench is in the fall on top of a mulched layer. This way the organisms will have food to break down, making it available to the roots of your plants. When you're spraying turf or lawn or even a hayfield, you can use either compost tea or compost extract. Compost tea has more microbial life in it, so that's why I'm going to be using it here on this space where I want to inoculate it with a lot more organisms than what extract would be giving me. The rate of application for compost tea or extract does depend on the biology that's present, but you'll want to make sure that you're putting down at least 5 gallons to the acre, or you could do up to 20 gallons per acre. Oh, good. Well, that's not something you hear at a composting lab every day. That's kind of crazy, you know? They call it black gold.
Elaine, is that your first compost pile? Do you feel like you just played a part in saving the world?
Think of all of the things in our municipal waste systems that have this potential to be a really valuable resource. So think about waste in a different way.